From Microbe TV, this is Matters Microbial, a podcast about the wonders of microbiology, microbiologists, and microbial centrism. This episode was recorded on June 27th, 2024. Hello, Micronauts, and welcome once again to our Quality Quorum for today. I'm your host, Dr. Mark Martin, Associate Professor of Biology at the University of Puget Sound in Tacoma, Washington. Today is the 47th episode of Matters Microbial. Those of you who watch and or listen and spread the good microbial word to others, many, many thanks. As I move toward teaching microbiology in the fall, a reminder that students are very creative. Last fall, I had Micronauts in my class create memes of concepts from that class. As you can see, the students are creative and clever. I look forward to this fall's class, the next to the last time I will teach microbiology before retirement. Eek! I received a nice note about the podcast from Greg. As you can see, he has not just kind words, but some suggestions. Thank you, Greg. I look forward to notes and letters. Greg also sent me this cartoon suggesting that none of us are really in charge, other than the microbes at the center of everything. From the time I was a young boy, seeing my first plate streaked with luminous bacteria, I was hooked on microbiology. I do my best to promote microbial enthusiasm in my classes, as well as the concept of microbial centrism. One way to do that is to tell new micronauts about what I call the heresies of microbiology, things that are unexpected to non-microbiologists. Ironically, one of the first heresies I teach involves how different microbes are in nature versus the lab. This first slide describes work done by our guest today. The second slide reiterates how different domesticated, lab-adapted, microbes are compared to their wild ancestors. And the third slide shows some beautiful images of domesticated and wild microbes in terms of their colony appearances on petri dishes. You see, Plutarch was right. The wildest colts make the best horses. Because of my interest in promoting microbiology wherever I go, it's a real pleasure to introduce Dr. Roberto Kolter, Emeritus Professor of Microbiology at Harvard University, past president of the American Society for Microbiology, and true apostle of the microbial world. Roberto, it is such an honor to chat with you. Welcome to our Quality Quorum.
Mark, it's really a pleasure to be here. It's wonderful to see you, even if it's only virtually. And very, very happy to be part of Matters Microbial today.
That's lovely. So you happen to be in an interesting place right now. Would you care to tell the listeners and viewers where you are?
I would love to. You know, since retirement, what we did with my wife, who is Colombian, we bought a little bit of land and built a house in the high Andean forest outside of Bogota. So I'm talking to you from our house in this gorgeous ecosystem in Colombia.
Yeah, it's just amazing that we can speak this quickly to one another as well, isn't it?
That is an amazing thing. You know, the internet here turns out actually to be, I hope, let's see, it doesn't get interrupted during our chat, but it actually turns out to be fantastic and better than what I have in Cambridge. And we are on solar power, which means we are not subject, hopefully, to what might happen in the next hour, a power failure. But hopefully the solar power will be there to keep us talking.
Oh, what you give me hope for my own retirement, though it will not be quite as nice as yours, I can promise you. What I was going to ask you, though, first, and whenever I have guests on the podcast, is kind of a thumbnail sketch of how you came to microbiology, just for the listeners and viewers.
Well, it's been my whole life, really, but it started, my interest was very much as a high school student on serendipity, perhaps, on DNA. Somehow it fascinated me, the mysteries. You have to go back 60 years to know where we were in the knowledge. And I love DNA. And so from there, coming into genetics as an undergraduate, I was enamored with bacterial genetics. That itself was all E. coli from the perspective of the times. And only slowly, would I say, did I enter the much wider realm of microbiology writ large. So that took a lot longer, but it is still going on.
It's so funny because I have watched, I mean, we overlapped a little bit when I was a graduate student and you were a postdoc long time ago. But it's so interesting that I keep seeing evidence of things that you've done as I present papers or have students do readings. You've had quite is footprint the right word, or shall I say flagellar print in microbiology?
Yes. It could be pilly print, I guess. But well, you're being very generous, but it's nice that you bring up our overlap because it is a transition period for myself, my postdoctoral training when we met, and I remember, in fact, you rotated in Charlie Nowski's lab, and so we actually, if I memories can always fool you, but if I remember correctly, we shared desks that were next to each other, and
we did.
You reminded me that we even shared some mate. In those days, I used to cebar mate quite a bit. So as a graduate student, I had worked on DNA replication, and it was a really exciting time to be doing that because the replicon hypothesis had not been really proven with any model system, and we were able to do it with plasmids. And so that was super nice, finding an origin of replication and proteins that acted on it. That was super nice. And I have to admit that by the time I came to be a postdoc, and that tells a lot about me and the science that I've always loved. The project I was working with, attenuation, to me, it was solved, right? But I had loved it when I applied to the lab because it was a mystery. By the time I arrived, it was no longer a mystery. It was alternative RNA structures. And to me, it was like, it's done. So it allowed me to explore other things. And I knew one thing for sure from the postdoc. When I would have my own lab, I would not work on the same thing. So that was really important. And when I opened my lab in '83, we were looking for things, and we found small peptides. We found iron siderophores, the whole concept of iron regulation. So lots of fields opened up to me. And since then, I've been very much curious about new things. And somehow my personality, or whatever you want to call it, leads me to do things that I hope not other people are doing. Perhaps because I'm afraid of competition. I don't know. The point is that maybe it is because of that this pilly print is perceived by others as large, just because I've explored many things. And I think that's something that people should say, that's what I want to do, I'll do it. If I want to work on one thing for the rest of my life, that's what drives me, that's fine. That's also okay. It's just that people should do what they really feel passionate about in terms of the questions they pursue in science.
There's a very interesting essay I read with the physicist Richard Feynman. And there's this person who's trying to make Richard Feynman become a mentor to him, and Feynman didn't particularly want to do that. And I don't know if it happened this way, but this really struck me, Roberto, and maybe it will you. The person speaking to Feynman said, I'm just not sure what I want to do. And Feynman says, I'm not going to imitate him. I met him once, and it's very easy to fall into that impression, and I won't do it. He really did talk like a taxi driver. It was amazing. But he said, go downstairs 2 floors, and they have a photograph of an atom. And this would be at Caltech. And they did. And he says, really look at it. He tells the person questioning him, really study it and ask yourself one question. Does it make your heart beat faster?
That's lovely. I had not heard that. That really is a moving anecdote. I love Feynman. He's one of my all-time heroes as a scientist, as a person, and his lectures are available. So it's really easy to become familiar with him. And I love that he actually said, go down and look at the picture of the atom, because and the concept of the passion and heart rate going up is wonderful. He also said, if you understand anything in biology, just look at it. So the concept of look at what's in front of you and then see what drives you. That's a lovely anecdote.
I think that it's also really, really accurate. I met him once, and I wrote a blog post about it. I'll send it to you, and I'll put a link in the show notes to it. He was a character, but This is how I feel whenever I look, say, at a flagellar motor, especially now that cryo-EM has become such a deal. You can actually look at them. You can actually see how things look. To me, it's just amazing. And honestly, there are many paths to get into microbiology, and we've talked about it on the podcast. There are lots of different ways to get there, and I really appreciate how you said if you really like one particular, and I'm going to use the word narrow in a non-pejorative way, but if there's one specific area that really drives you, follow it. Or if you have many broad interests, find a way to follow that. And I think that that's exactly right. As someone who teaches at a primarily undergraduate institution, and I have a little lab here, and while I don't have grant support, I do have a lot of curiosity and small dollops of money, so students can look at all kinds of things. And it's really, that feeling that you surely have, like, I didn't know that. There's nothing better as a scientist than that.
Absolutely. And these days for me, a walk in the forest, it's not quite like as exciting as my dogs seem to be whenever they get a new scent, but whenever I see something, a new lichen or a new mushroom sprouting, I'm just fascinated. I want to know more about them, right? And it might be that somebody looks at today at lac repressor binding to DNA and says, hey, I want to know more about it, right? It's just the questions that arise when you see something, and that I think will. That's not just for scientists; that's for everybody. I mean, I think this is curiosity drives a lot of how knowledge is gained. So,
it's funny how we call it childlike, and if you have childlike curiosity, people say that in almost kind of a negative way. But honestly, that's how I look at most of the world. I mean, something I didn't know before. That's interesting.
If we are fortunate to keep our childlike curiosity, that is a blessing.
It really is. So, Roberto, you set up your lab. You looked in a couple of different areas. Are there particular types of things that you'd like to tell the listeners and viewers that really stick out in your mind? Before you do, I want to say parenthetically that I use several articles that you've authored for my courses. And there's one, I believe it's Frontiers in Microbiology, and I'll put a link to it in the show notes. And it's this wonderful business of how you got back to ecology from microbial genetics. And I find that whole thing fascinating. What particular things do you think might spark interest in our viewers and listeners from your work at Harvard?
Oof, that's a wonderful question. Food for thought. Let me just tell you, I think that the article that you're referring to is International Microbiology. It's the journal. It's called A Voyage,
yes,
like a molecular genetic voyage to microbial ecology. And I know it because it was a lecture I gave when I was president of the ASM at the Society for Microbiology of Spain as an invited guest. So that's why I remember that International Micro is the official journal of the Spanish Society for Microbiology. So that's the and let me just say, of the things that strike me that I would like to talk about what I did over 35 years having a lab at Harvard, which is tough to compress into 2 minutes, but I'll try, maybe 5. So I think that the nicest experiments that were carried out in my lab, and this is always dangerous because the 100 people that I'm not going to mention might take offense, but that's not the point. I think that the ones I loved the most were the simplest ones. That is the ones that you had no idea what the answer would be, but that you could carry on with extremely simple approaches. And so, number one, when we noticed that actually the majority of cells in the stationary phase culture were dying, and the conditions that we were using, I won't go into the details, but death was very fascinating. But then the question was, what are the guys that are surviving? Are they adapted? Are they E. coli spore-like things? And so then the simplicity of the experiment that we did, we took some guys who had survived after 2 weeks, and we put them back in at overnight, see what happens, how they react. And the fact that we saw them take over, so that immediately said, geez, this is evolution happening in a matter of 24 hours, right? A whole population takeover. Wow. it took us a long time to convince ourselves that they were mutants and not just adapted strains, adapted cells. But that was a fantastic experiment. The other one, the simplicity and this of when we started working with the biofilms and trying to do genetics in biofilms, and the concept that we could just grow things in 96 well plates and then stain the walls of the plates with Kumasi blue and see the ring that was a biofilm. And that means if you could get mutants, you wouldn't see a ring. The power of that simplicity, and then to see that thousands of other people went ahead with that was great. And then I'll say, go ahead.
What I was going to say about both of those things, the growth advantage in stationary phase, the GASP experiments with,
yes.
Come on, you were part of Life After Log. That's a great title. I'm doing an experiment like that right now while we're speaking. And it has had such an effect on my students to the point where they tend to ask me questions. Are cultures really homogeneous? And I'll say the real question is, are colonies homogeneous? And the answer to both those questions are no. And it's horrifying to them. And I say we have to start with very definitive ideas, and then we get to find out. It's like the physicists say as a joke that constants aren't and variables don't. And it's funny to a physicist, but when you're studying, it's horrifying. And it's really true. I mean, it's part of the natural world, and variation is key. And I find this fascinating that you can see these things taking place, which is wonderful. As far as biofilms go, I needed to have a project where I taught students about data manipulation in microbiology. So I would just use 24 well plates, Pseudomonas putida, and they got really wonderful data that they could analyze. And then I found that alpha amylase actually inhibits biofilm formation, which gave them some thoughts about what the glycolix or the adhesive matrix it actually is in a biofilm. But the part that you would like is when you use amylase, you can pick up the whole biofilm from the well.
Absolutely,
which means
great.
what holds it to the plastic is different than what holds it to each other.
Mm-hmm.
And this takes it back to your idea of simple ideas and simple observations. This is the best piece of equipment in the lab right there. Well, maybe not this particular one, but the students doing things. And I tell them, use your look at things, really think about it. Does it make your heart race, right? And it's really critical for them to do that, because the number of times that people say, this is amazing, I hadn't seen this before, is fascinating. So and it reminds me, I don't know who wrote the old genetics paper, Logic in Toothpicks. I want to say John Roth, because it sounds like John.
It could be that, or it could be Josh Lederberg. I don't know. I don't know.
But the point is, as you say, simple questions, because the world is complex. And if you can distill things down to where you can study them simply, how wonderful.
I agree.
I
want to emphasize one thing you said, and that is that in the end, no matter what machine you're using, the observer is the key, right? So you have to rely on this. The observer, you can use, and I actually recommend use as many techniques as you can, or as necessary for the question you're asking. But remember that the observer is you, and it is your mind that is doing the analysis. So without a doubt.
Oh, yeah. So it is very clear to me that the way that you set up your lab is you encouraged your colleagues to look in different directions. And it really paid off in many ways, as I look over your CV in particular.
Yeah, there's one thing that I took very much to heart, and that was I felt that it was important for everybody who trained in my lab to have the option to continue with that particular line of interest, and that I would not represent a particular competition going forward. Because I liked going into new areas, that was very much consistent with how I love mentoring, thinking about new ways, new directions to go. So in the end, it was a model that for me worked very well. And I actually encourage people to do it. People are a bit afraid because sadly, they are quite interested in the area of comfort. And I think that has to do with the funding system, etc. I somehow managed to run a lab, I think reasonably successfully, without worrying about this. Am I satisfying the aims of my grant? That is, my curiosity was what drove the research more than what I had written the specific aims for the grant 2 years ago. And usually we did okay with satisfying the requirements of funding agencies. So I do recommend it as people to not be so tied to the area of comfort. But you know, it varies with persons.
Absolutely, and there are many paths. I don't know if this is an improper metaphor, but I like the idea, rather than a big tent, a big petri dish for microbiology. And in particular, people who come to things from outside formal microbiology often have really interesting ideas. And that cross-fertilization is important to me. The very history of molecular biology was founded by quantum physicists who didn't like working in quantum physics anymore. So that's
right,
fascinating to me.
Yes, I love that history, and I love the whole history of our field. So that's something that I wrote a little essay that you may want to put in your show notes on the history of microbiology. So I think
I'd love to see that.
that is and when you say the physicist going in, that I like the old phrase that this is at the time in the 1940s. This is the physicist leaving the science of death to enter the science of life. This is the people. They did not like being part of the Manhattan Project anymore, and that was a great incentive to go. What is life? This is Schrödinger pulling them all. Let's analyze life. And so. Anyway, I don't know how we got into that, but there we are.
Well, no, it's where people are following their interests. I mean, as you mentioned, Leo Szilard is a great example of that, who invented the chemostat. I had no idea, but there you go.
Let me interrupt you with that, because those guys were Novick and Szilard, and their chemostat and periodic selection were such an inspiration to us when we began to look at these takeovers in stationary phase, right? Yes. The gas mutants. And that is something that I really have to emphasize, how important it is to look back at what others did before, and not just 5 years ago, but really go back and see, because a lot of stuff gets done and then gets sort of forgotten, which is too bad. And I think is why I believe so strongly that the history is so important.
Oh, I agree with that. And I get a certain degree of pushback on that. And I have to say that I'm going to start collecting a number of things that were discovered nowadays that were reported in the 1930s and 40s on all different kinds of levels. It's just, you have to keep your mind open. I mean, the very discovery of what you and I call quorum sensing probably had to do with natural transformation in bacillus. And that was Novak's work in the early 60s. With conditioned media seeming to enhance that process.
I have to say, yes, don't get me going on the quorum history, because I mean, definitely the competence factors that were being discovered in the pneumococci and the A factor in the streptomycetes greatly precedes luminescence. I mean, everything had point. In fact, people were saying there are these things that are density dependent. Anyway, so that's, but talking about things from the past that are driving ideas of the future, let me just tell you some of the something that I think is really wonderful that I'm now beginning to work on, which is abortive transduction. And this is something that was already discovered in 52. As soon as transduction was discovered by Lederberg and now I'm blanking out, Zinder, in 52, Bruce Stalker, who was working on motility, immediately went to work on transduction. He wanted to do the genetics of motility in Salmonella. He was always interested in flagella. And he discovered this concept of abortive transduction. And what that is simply that the majority, like 10 times more than transducing fragments, are these fragments that come in, chromosomal fragments that come in, circularize, and since they're not replicating, that is not a fully transducing event, so that's why it's called abortive. But that piece of DNA stays in that cell. When the cell divides, only one of them inherits it, right? But we have not considered the role of abortive transduction in current studies of microbial communities. So, as this picture shows, uh, what might actually be happening in a community is that cells are not just having their own genome, but they are testing fragments of incoming transducing particles which are not replicating, but are changing their phenotype, individual cells. And if you think of this as a community, this is now many cells testing out the DNA without necessarily incorporating it. And the ecological consequences of that, I begin to marvel. So turns out I have the luxury of being invited this summer to do just in a couple of weeks. We start in Santa Barbara at the Kavli Institute of Theoretical Physics, where I'm going to be leading a lab that is testing the idea of transduction into non-growing cells. I think that we have forgotten the non-growing cell as a key point in the physiology, ecology, and evolution of natural communities. So I'm really going back to my roots of stationary phase and taking a principle that is from the 50s and seeing, and I'm really excited because all these physicists are going to be learning how to do transductions.
And it's simple enough that I could do it, and that means a physicist could do it because I did my share of P1s. And so this is going to work very well. I really like this idea of coming back to old ideas with new sets of eyes. There's your next essay, right? And
That's right.
and it's a wonderful idea. I had a student in my lab who, for a project, and I'm trying to write this up. I got a hold of some lambda-vir. I hit up some cells with lambda-vir. I look for the survivors. I look for the ones that can no longer use maltose. And then we use PCR to fish out the LAMB gene and look for the mutations in it. And this drives home the idea of how evolution is taking place all the time using very old papers and old ideas.
Absolutely. I think it's great.
Yeah,
I think that there's very simple experiments that drive ideas home. And I like very much this, that the future is sometimes in the past, right? So
it
we
is.
just said,
That's Gabriel Garcia Marquez said that. I have to find out where that's from. But yes, that's a really good one. Oh, but oh my goodness. A novelist, he was a Southern Gothic writer. I will get you the actual quote, Roberto, but it's like, the past isn't dead. It isn't even past. And that's 100% true. We have so much to learn. And when you have introductory students the way that I do, because I only have one microbiology class, they can understand papers written from the 1970s back. Because there
yeah,
aren't
well,
such...
some of them are really quite fascinating, but and they are difficult to penetrate. But when you do, because the genetic logic is so beautiful, when you actually, it's like, oh my, the cousins, Crick's cousins, and the phage experiments that led to the triplet code. I mean,
Yes.
that's a beautiful paper, but it takes some explaining.
Yeah, it's like a
But yes,
McClintock paper. Yeah. No, this is quite wonderful. And your idea about non-growing populations, because the majority of microbes out in the environment are non-growing. And
yeah,
so this
it
is...
is a fascinating thing. The doubling rates is something that I'm very excited about, but I think that you're right. Most of them are not growing fast. And my belief these days, based on, but it is a belief, it's based on very little evidence, is that the rates of horizontal gene transfer are greater than division, so that an individual cell can actually experience multiple horizontal gene transfer events before it duplicates in many settings. And so we really don't have an idea, we don't have a clue of real frequencies of recombination as a function of division. And I think that really will turn it's the way we think about microbial evolution on its head, if that turns out to be the case. So part of what I'm doing is sort of aiming at getting quantitative estimates of horizontal gene transfer
And
in
physicists
non-growing
are good at
cells.
that. Physicists are good at that. So
Yeah,
this will work out
exactly.
very well.
Yeah.
This reminds me a little bit of Carl Woese's idea that there was a period of primordial history where horizontal gene transfer was everybody like sharing DNA all the time, doing everything. And that's what I was thinking about. So maybe those were the earliest forms of life, were things like biofilms with very primitive cells that are just constantly sharing genetic information. And I love this idea a lot.
Right. And I think now the huge race that we're seeing are what is probably blocked by recombination barriers, but within what we call closely related species, I bet the rates are just enormous, which is why we see this remarkable diversity in strains.
Yes.
And so the rates have to be really, really fast of the sharing anyways.
I'm getting too excited about this. So, which, if you don't mind, I'd like to shift over a little bit to your work in trying to promote, I'm not going to say microbial literacy. How about you're promoting microbial enthusiasm to the public?
Yes, I would love to chat a little bit about that, because it is a huge part of what I'm doing these days. I'm now taken over from Elio, Small Things Considered. So blogging is something that I do, and that's still for a rather, let's say, already microbially enticed audience. They have a reason, but I do a lot of stuff also for the public that may have never heard of microbes and might have a certain view of microbes as limited and pathogen centric, etc. So I've been doing that for a long time, and I just like it because I think microbiology is beautiful. Microbes are beautiful, and so there's easy ways to capture people's attention so that the curiosity can arise and they can ask questions about what's going on. So let me just say, I'll bring up, it's not necessarily advertising, but maybe it is. Here's the book cover of Life at the Edge of Sight. And it just says how fortunate I was that Scott Chmielewski came into my life. So around 2015, as I was already thinking about closing my lab, Scott walks in, and we got along. He's a photographer, and he's also a very talented writer, a wonderful man, and we just hit it off. And within 1 year, we proposed to Harvard University Press this idea of the book, and we produced it, and we handed in the rough draft. And so it is, I think, a testament to Scott's abilities to do the photography and then also the writing, which we did together, and that started a whole series of events that led, for example, to what I consider beautiful exposition of photographs. And my favorite is actually not a photograph. The exposition is called Life. Hold on a second. It's called A World in a Drop. A World in a Drop. And the idea is how much of microbial life is inside a drop of water. We call it teeming with microbial life. In a poem that we wrote, actually, there's a poem that accompanies the exhibition. And but my favorite part of the exhibition is this exhibit of a 3D print and I'm showing it to you here, this 3D print of a Bacillus subtilis colony, which was hung in a dark chamber. It's about something like this wide, 8 inches or so in diameter, and it's illuminated. And the inspiration came from the Colombian Museum of Gold, where many pieces are exhibited that way. And I just thought that this dark background and this shiny colony just was so beautiful and so captivating. People had to say, what on earth is this thing? And to begin to look at the wrinkles and ask questions about why the wrinkles, etc., can lead to lots of answers. This other image in that World in a Drop exhibit is actually it's a micrograph that I'm showing you here, differentiation and the division of labor in the Bacillus subtilis population, where you have different cells expressing different genes. And so in this particular image, the blue ones are expressing surfactant, the yellow ones are not dividing, they're making filaments, they're making a matrix that holds them together. And in fact, they need the surfactant made by the others to migrate and to conquer territory. And this is work that was done by Jordi van Gestel, who is from the Netherlands, and inspired by Jordi's origin. And we were just talking, this looks just like a Van Gogh painting. And so we actually called these bundles of cells the Van Gogh bundles, and that's what they're called in the publication. So there's this beauty that we can now show in museums, and this started at the Harvard Museum of Natural History. But this particular World in the Drop exhibition has been traveling all over the world. It's been in China. It's been at Caltech. It's in permanent exhibit at the Danish Technical University. It's been to Chile in various locations. In Uruguay was actually, I have this photo of the exhibition in Uruguay in the street. So, it is just so nice to see microbes so that people can walk. I've always been fascinated by these photographic exhibits on the streets. So seeing our photographs on display in the streets of Montevideo, what a wonderful, wonderful thing to witness.
Let me just say, if you don't mind my jumping in, my wife loved that 3D print of Bacillus subtilis, and Scott was able to give us the STL file. And so as you're looking at now, she made, because it's for me, things that are fluorescent green and fluorescent blue copies of that, speaking of beauty. And now I actually like your idea to kind of like the old, like Incan or Mayan type of display is probably a little better, but I like things that glow in the dark too. It's kind of an issue of mine. But how wonderful that is. The second thing is, I am not well traveled, but going to the Van Gogh Museum in the Netherlands meant a lot to me. So I'm going to have a really careful look at that picture because I need some new artwork, and that would really fit very well. I also want to tell the listeners and viewers, in that image that you showed, it's possible to use a technique to see which genes are active at different times. The different colors are showing that, and that's how you're able to visualize the division of labor. And it's funny because you talk to most people and they'll think that all the cells are doing the same thing. And I point out reasonably, your cells aren't doing the same thing all the time. So there you go.
That's right. And bacteria can do the same. And this concept of populations coexisting and actually cooperating by expressing some population, expressing some genes, others expressing another genes, they can do much better evolutionarily than if they try to express everything in a single cell. So that's division of labor. That actually has this evolutionary advantage to it. So, and it's wonderful that we have techniques in which we can turn a particular fluorescent protein on by a promoter that drives genes that are, in this case, making surfactant or making the extracellular matrix, this exopolysaccharide.
I think I've seen a video of this as well. And I show it in class, and I'll try to find a way to put it in the show notes. And what's interesting is you're able to follow things in real time. So there are cells that turn on a set of genes, and the same cell then turns it off and turns on the second set of genes. Again, yes. The power I never would have guessed in graduate school that you could use.
Yeah. So that's what I was saying. These techniques come up, use them to answer the questions that you have. In the end, you will be observing the results, but the techniques, as they come, make sure you use them if they're appropriate. I mean, it is. Don't ever be feeling limited because the technique is out there, but I don't know how to do it. Well, go learn it, right? It's as simple as that. Go find somebody who will teach it to you.
So you blog, you write, and you're doing outreach via, I'm going to call them museum exhibits, and all of this is wonderful. Are there other areas which you feel you could be promoting microbiology to the public?
To the public, I get occasionally asked to give lectures to the public on science communication sometimes, and so I do that. I do that with great delight, and I do lots of lectures now. Of course, a lot of them are in this virtual setting, but also by going places. So lectures, I think, are still, to me, is extremely, I tell them, they're energizing to get in front of an audience and talk about what you love. And sometimes the audience is very much microbiology, but sometimes it's just students at the university, general audiences. And I love that as well. Yes, it's interesting, I closed my lab, but I never meant to slow down terribly, which I haven't. And so I love it. I think it's an opportunity that comes at a time when I was really ready to close my lab and just go out and do other things. And it's been wonderful. So it's been now, I think, I I I, I don't want to say it without thinking about it. So because yeah, almost 6 years since I closed the lab. So it's been busy 6 years, even though we for 2.5 of them we were in a pandemic. But that's all right. I was locked up in the forest, so it wasn't being locked up.
You mentioned finding ways to promote the public's interest and some of the sculptures. If you, I believe that the 3D print that you have of the Bacillus subtilis colony is in metal. Is it not in
Yes,
metal?
yes, it's printed in metal. Yeah, yeah. So that
Yeah.
that is the color. I have to say I don't remember what the metal is, but that is the color of the metal that was used for the 3D print. It's
Yeah.
not it's not false colored.
So it's a wonderful way to kind of promote ideas. And I've done some of that with some of the bioluminescent bacteria. You must have seen some of the Lux art that
Yeah,
we
yeah,
do. And
sure,
I'm useless with it. My wife's a fabulous artist, but I found that when students give the opportunity to do this, it teaches them that not all bacteria are bad. Yes, I hate that. Oof. I have a whole lecture I give on that. And then secondarily, that there can be beauty involved with it. And it's a funny kind of beauty because it kind of fades after a couple of days. So I think it's wonderful. And I'm looking for things like that to promote. I know Dave Westerberg does stuff like that with different types of, I guess he has an indigo producing bacterium that's light responsive. Same principle. So the whole thing is putting things in the hands of people to try. And I think that's a form of outreach. And that's
sure.
something, I might find a way to do in the future. Who knows? But I certainly am thinking about what I'm going to do in just a couple of years. And you've shown me a decent path here. I really appreciate it.
Let me tell you another path that I'm sure I'm not the only one following it, but fermentation. So I think fermentation. What a better way to get the people's curiosity about microbes than actually making delicious food. And so thinking
Mm.
about that, Pia Sorensen and I made a course, an online course, which is extremely accessible. It's actually, in fact, if you ask me, maybe a little bit too easy to access microbiologically, but that's okay. It's actually turned out to be a very popular course in Harvard X. That this is the online course portal. It's free, so I recommend people glance at it. So it's a really nice way to reach out, and so I'm even thinking of doing it here to my local community. We have an organic garden that is for the whole community, so it's several acres in size, and of course we always get too many cabbages, right? So why not teach them how to ferment and other things, and making our own beer, and all of that, isolating yeast from the forest and making locally brewed beer from our local yeasts, things like that. So there's always things in the planning, and I think fermentation is really a great one, because what a better way to express microbial activity than in food that you can take delight in eating afterwards.
Absolutely. And there are so many different types. When I would start thinking about that, I was amazed by, I mean, when you talk to them about alkaline fermentations and for the Japanese delicacy natto, which
Yeah, yeah.
is not to my taste, but
Yeah, I
perhaps you
must
like.
say, I worked Bacillus subtilis for several years. And the thought of eating stringy natto is still not my favorite, but they love it, and so that's okay. You can
Yeah.
make natto.
Yeah.
Yeah, yeah.
And and there have been so many, and in fact, I'll put a link to some books on fermentations that are very accessible to the public that I think will be good. I recently had Rachel Dutton talking about
Yes,
cheeses on the podcast. Fabulous subject. You want to talk about
absolutely.
stinky microbes in the lab, though? That was the case there. So
Yes,
so
they
may,
can be.
so in some ways, I wonder if it's possible for me to maintain a small lab footprint to do a few things that interest me. And that's something that I've been doing a certain amount of thinking about. And you're doing things like that. Are you doing that more formally? Do you have like a little lab that you're creating, or are you just traveling from place to place and using their facilities?
So currently, all that I do is outreach by saying, "Hey, can you please start a culture now in your lab and then have it shipped to Santa Barbara? Because I don't have the capacity to do it here. However, there are some things so that I think are doable. So my wife and I, she's also a microbiologist, so we are setting up here a small lab in our in one of the rooms in the what we call our secondary kitchen. It's just a back room, and we are going to be doing some isolating of because we've been very interested in the communities that are living in the lichens. So lichens are now a fascination of ours, and she's shown from her own work that there's a core community that is present in all the different species of lichens. So we want to work with those. And I should say this is actually completely crazy, but also going back to our gasping studies, because I'm now beginning to interact of all the. This is really neat. I'm interacting with an architect, a really wonderful architect, professor at MIT, Carlo Ratti, and he's an urban architect. And he came to me because he read Biofilm City of Microbes. This is an essay that we wrote. It's a bit like Life After Log, but this was on biofilms. It was called City of Microbes. And he was fascinated by that. And now we're doing lots of work together. But one of the works that I'm doing with him involves for Jeffrey West at the Santa Fe Institute in scaling. So here, this gets pretty crazy, but I think there's some merit to it. And that is, are there principles, population principles that apply across all scales, from microbes to humans? And so this may sound crazy, but what we're trying to model is predictions of when a population behaves a certain way as it's growing exponentially. What does that tell us about what it will do in stationary phase? And you know where this is going. You know, there's 10 to the 10th bacteria in an ml, and there's 10 to the 10th humans in one planet. And how can what they have done in the past predict what they will do in the future? Will there be catastrophic collapse? Will the coming population decline be orderly? And can we predict that from models of microbial growth? And it might sound crazy, but there's enthusiasm among the three of us to think about this. And Carlo, as an architect, has been named the curator of next year's Venice Biennale. And that is this remarkable exhibit in Venice that 1 year is art, modern art, and 1 year is architecture. And so we're going to put microbes next to cities. And we already gave a talk together about the similarities between microbial populations and biofilms and human populations and cities. So stay tuned for the Venice Biennale 2025, where microbes will be next to humans in a really very important worldwide exhibit. So that's got me really, my heart beat racing.
It's interesting, too, because when I would have students make little miniature Graski columns, I would call it planet and then their name, because they put it together. And in a similar way, I know Scott had made an enormous Graski column.
Yeah. We had one at a museum. That's right. That was used after the World in a Drop exhibit. We had a microbial life exhibit that ran for 3 or 4 years at Harvard Museum of Natural History. That had a beautiful column that we put together. That was, it's more than 7 feet by the time it's on this pedestal, right? So it must be 5 feet tall and maybe a foot in diameter. And it was just absolutely stunning in its color development, because we had lights, grow lights on it. It's actually, when the exhibit was closed, it was moved and currently lives in what is called the Northwest Building at Harvard main campus in Cambridge, and it's still there. And so when we have the yearly Microbial Sciences Symposium, which is held down there, there it is. The VOG Graski column is for everybody to watch, and it's really stunning. It's still turning all sorts of colors. Now a little less vibrant because the lights are further away.
You know, you can actually make microbial fuel cells with them. And I've often thought that that's a really interesting idea to watch how that changes with time in an art exhibition as well. You could even imagine connecting that rather than having a blinking light, having small amounts of sound and watching how that sound changes with lights and nutritional values. That would be like a lovely art, kind of a living art kind of thing as well, wouldn't it?
Now that you mentioned this, it's great because at the Microbial Sciences Symposium this year, there's this person, and I'm going to have to send you the link. This person who has a shop that is just full of vessels that are doing just that. He's got different microbial activities producing all sorts of sounds and noises from the bubbling and from the electricity, etc. So you walk into this, and it's a little shop, but it's like walking into this fantasy world of microbial activities that are perceivable in the micro scale, right?
I think you've just identified my future with that. So that'll be fun. I can't imagine what my wife's going to think, but she's put up with me for so many years. I'm sure that'll be no big deal. So Roberto, when you're mentioning the business of architectural views of humans versus microbes in groups, cities of microbes, cities of humans, are there emergent properties that develop, certainly for the microbes, say in biofilms or in colony development, that can be mathematically modeled? And would that be applied to perhaps some of the urban planning that you might see in human populations in human cities?
Well, that it's fascinating you asked that question. And immediately what comes to mind is this wonderful slime mold, Physarum polycephalum. And this is the single cell that will go out seeking food after it germinates from being a spore, and it goes this paths and it's seeking paths for food, and it optimizes the path for food, and it actually communicates within the single cell to retract the pods that have been searching and where there's not much food anymore. And the key point here is that you can use mathematics to model the behavior of this and recognize that this little organism can uses the optimal way to communicate across its paths. And when people say, let's model what would be the most efficient way to make mass transit in a city, you use the Physarum approach to optimization to optimize movement of people. So there's a place where mathematics comes into place, modeling comes into place, and computer science comes into place, because you will develop these rather complex models that computational systems can predict the answer. So it all plays a role, and in terms of the scale that you go from, it's fascinating because this Physarum is really brilliant, and it will solve mazes. It will remember the maze, and the best. It knows how to optimize to the best path to get the maze solved. So it is a thing that you can even say you can model from the behavior of a microbe to the larger scale.
The small teaches the large.
In that case, yes.
Roberta, you were talking about opening eyes. And there's a story that I've told before, and I'm going to tell it to you really briefly. About 1 million years ago, when I gave a formal talk about bioluminescence, I had an old Fernbach flask. And this is before we had cell phones, so it was actually really hard to take pictures of bioluminescence. So I had like 50 people in the audience. We turned the lights completely off. I swirl it and hold it next to my head because it looks really creepy, right? And there was a high school student in the front row, and he didn't know that formally you're not supposed to raise your hands during a seminar. He didn't know that. I'm not saying that's good or bad. He just didn't know.
Good for him.
And he raised his hand, and I wasn't put off at all. I mean, why would I be? I'm not threatened. And he raised his hand. I said, "Can I help you?" And he says, "When you hold up that flask, I can't see through it." I said, "That's true, because you have a fairly dense culture of bacteria there." He says, "How does the light get out?" And I'm told I sat on the floor and thought about it. And I believe it. It sounds like me. And what I told him is this. I said, "Now, I've never been asked a question like that, and I never, ever want you to change. I want you to ask questions like that, because too many times we limit the questions we ask based on what we think is, quote, a smart question. Oh, I hate that. Or, quote, a good question. Questions are questions. And almost certainly if one person has it, other people do too. But what I told the student is, if you imagine, and this is quick thinking on my feet, I guess, if you think of a lamp with a lampshade on it and you don't turn the lights on, you can't see through that lampshade. But when you turn the light on, you can see it. And I said, perhaps the cell wall of the bacteria is like that lampshade. And he says, that makes sense, and sat down. And I was really struck by that because he wasn't threatened. He asked the right kinds of questions.
Mm-hmm.
And that's your and my goal, I think, is to make sure that we encourage people to ask those kinds of questions.
Yeah, absolutely. And actually, that immediately, that question, it isn't just about microbes. It's a question about the physics of light. And,
yes, it is.
And what causes it to be, what causes a culture to be cloudy, what's happening to the light, and that brings up the whole concept of scattering as compared to just transmission. And so I think this is where curiosity really drives discovery.
And when I've told people that story and I take a pause, I will get people who say that's kind of a dumb question, but it wasn't, was it?
No, not at all.
And that's the thing. We need to move away from that idea and actually kind of, does it make our heart beat faster?
Yeah.
Right?
Well, one of the rules that we always say in the courses that I teach during the summers, it's there's no such thing as a dumb question. Just instill in the students that there's no such thing as a dumb question. And they'll still say it. This is really a dumb question, but okay, well, ask it anyways. There's no such thing as a dumb question.
I think that what they mean when they say that is they want certitude. And I understand that, but that's not dumb.
Yeah, exactly.
I just think almost always when one person has a question, other people do too. And in classrooms, I've had a person ask a question and then someone else will answer it for them. So it brings everyone in.
I think you're fortunate in terms of the teaching that you do, because you're dealing with undergraduates and their curiosity tends to be less jaded and less focused. And so by the time, every time that you get more and more into something, then you get less and less open to asking the really naive questions that are sometimes the most important ones.
Well, I always tell people that I'm a giant goofball, but I aspire to be microbiology's giant goofball. And so keep that enthusiasm, keep that whatever you want to call it, have that enthusiasm for what you're interested in and keep pushing on it.
I agree. Go for it.
Roberto, this has been such a fun time, and I'm going to put lots of links into the show notes. Do you have any last words of wisdom for our listeners and viewers?
No, Mark, I think I just have words of gratitude. I think I will be grateful for everybody who I had the pleasure to interact with over these years. And I'm really grateful to you for asking me to this podcast. I applaud the work that you're doing. It's really great. And it's also super nice to see you after just a couple of years, since 1980 or 81 or whenever it was that we overlapped at Stanford. So it's been great fun.
My favorite Charlie Yanofsky story, rest his soul, I was petrified of the man. Just petrified. He was very brusque.
Most people were.
Yeah. And he asked me to look at something involving Neurospora crassa, and it just didn't make sense to me. And I thought, well, Charlie asked it. He's a smart guy. And I just knocked myself silly trying to figure it out. And finally I went to him hat in hand and I said, Charlie, I'm so sorry. I couldn't find an answer to your question. It didn't make sense to me for this. And he says, I said that? That wasn't very smart. He says, you shouldn't believe what everyone says to you. And that's actually pretty good advice.
Yes, it is great advice. He was a very wise man. I absolutely loved Charlie. He was really, really brilliant man. And it was a wonderful time to be there, even though I didn't particularly love my project.
Yeah. Well, I hope you and yours have this wonderful time. I can't wait to see what you're up to next.
Well, thanks a lot. And it was wonderful seeing you.
Thanks
Bye-bye.
again, Roberto. Bye now. This has been Matters Microbial, a weekly podcast about the wonders of our microbial world and the people who study it. You can send questions, suggestions, or comments to me at mattersmicrobial@gmail.com. Show notes from today's episode, as always with tasty links worth your time, can be found at microbe.tv/mm. If you like our work, please consider supporting us at microbe.tv/contribute. I'm Doc Martin, and you can find me in the biology department at the University of Puget Sound in Tacoma, Washington. Dr. Roberto Kolter is Emeritus Professor of Microbiology at Harvard University in Cambridge, Massachusetts, and parts all across the world. Many thanks to David Renata for superb editing and Reber Clark for the wonderfully quirky music that brings a smile to my face. I hope that you've enjoyed being part of our Quality Quorum today. See you next time on Matters Microbial.